Hey, beer appreciators, Sawadee Krab. Welcome to Beer Time Story, the platform where beer and brewing knowledge is shared. For today's session, we will have some good burps about bound thiol and its liberation capability from engineered yeast. I know that the sentence that I just mentioned contains a lot of sciencey word, but believe me, this session is worth listening to. Now, speaking of thiol, we, Beer Time Sorry, maybe a little bit late to the game, but it's better than never, right? And if you're speaking about or speaking of thiol in beer brewing, it might not ring the bell for beer consumer, but for the brewing industry, wise the one who make beer, it is considered to be a hot word in the past couple of years. There are many studies and research, especially from various yeast labs, that look into the realm of thiol and how it can possibly impact brewing processes as well as the final beer in your glass. One of the interesting aspect of area is how one can harness the power of thiol compounds, and there are many approaches to this question. Which one of the unique methods is by using an engineered yeast, and one of the labs that stand out in this arena happens to be the one that our guest speaker works at. So please welcome our guest speaker, Dr. Laura Burns. Hi, hello, doctor. How are you
Hi.
doing today?
Oh, doing wonderful. I'm so happy to be on the podcast this morning with you.
Okay, welcome to the platform. What's happening in Chicago? Like, how the weather is like, and all things.
We're in the peak of summer, and it's just beautiful. I have a big vegetable garden, so I'm just enjoying all of the wonderful tomatoes. It's really nice here in Chicago.
That's good. I heard that the city's nickname is Windy City, right? Windy
That's right.
City?
Yeah.
Okay.
Yeah,
Cool.
Partially because it's windy, but partially because of the politics. Things get brushed under the rug, apparently. Yeah, so that's how the name came up.
All right, that's good. Good to know that, as it has another kind of meaning too.
Yeah.
All right, so okay, just to warm up, our conversation a little bit before we hit the main road. Are you currently working on any interesting project or study that you can share with us at Omega Yeast?
Well, yeah, I think a lot of our work is continuing to, find really, additional features that yeast can add to fermentation, what ways we can improve on kind of either the quality, performance, or even, like aromas as we are with thiolized yeast.
Right.
But mainly we're looking into just novel strain development, bringing new advantages to yeast and allowing them to, make life easier for the brewer.
Okay. Thank you for that in advance.
Mm-hmm.
I'm quite surprised to find, doing the research of this session, I'm quite surprised to find that you are also, I'm not sure are or were, but you also have a passion for kombucha brewing, right?
Yeah, actually, and sourdough bread and any fermentation, pretty much. The spark of my passion in brewing came from really overall interest in fermentation. So throughout kind of my home hobbies, I've always been, pursuing any weird fermentations that I can do at home and experiment with. It's always been kind of the, more the art of it for me, because I love the science, but it's fun to apply it and make something kind of more, a little less controlled, but also, really just bringing kind of the science into cooking or into my kitchen.
Right. I think I have a little bit of knowledge and experience in kombucha making, because my mom, at one point in time, she was kind of like, she want to try doing it, and I have to assist her, order stuff. And with some brewing background, we know that you need to have some food source for your microbes to survive, right, and produce you those delicious drink. But the first time that I ever saw this SCOBY, I was like, what is this? for kombucha?
Totally.
Yeah. That's amazing, but it
Yeah,
turns out great though.
yeah, and it's a co-culture, so it's quite difficult to pin down exactly how the culture changes over time and the exact composition of that SCOBY, but the SCOBY can vary in composition even and make different flavors. So there's a lot of science to learn about kombucha making, and I know there's a lot of people working on that too.
Right. And last one is, do you still keep this hobby at home, or you just shift gear to the bread making instead?
Mostly to bread making, but I still have all my kombucha kind of ingredients, and sometimes we even at work here at Omega we'll share a little bit of the cultures between each other, and so we've picked up kombucha here and there because some of the employees here are also interested in making it.
All right, that's cool. And by talking to you with kombucha, I think I have to kind of find a way to just making some of them sometime soon. Okay.
Awesome. Yeah.
Okay. All right. I think we are ready to hit today's topic. We have a lot to tackle. Bound thiol. So, Laura, I think let's begin with these simple sets of questions. For our brewing world, first, what is thiol, and where can it be found, and why should one care about it?
Sure. I love the slides here on the side. They're very helpful, but they're basically compounds with an SH group or sulfhydryl group, and they're commonly kind of found across a wide range of natural kind of fruits. So any of the characteristics we describe as thiol aroma in the beer is actually the same compounds that are found in the fruits. So if we say grapefruit, that is the 3-SH compound that we're recognizing in grapefruit that's also found in beer. So across kind of tropical fruits, the ones that are primarily described for as aromas. So another example would be rhubarb. There's quite a bit of thiol content in rhubarb. So the link between those is truly the compound itself, the 4-MSP. So you can find it across a variety of plants, including hops. So the free thiols that are mainly in beer without these yeasts that we're going to talk about today are coming from hops themselves. And that's what brings more of the tropical aroma. We get a lot of fruit flavor from hops, a lot of dank character from hops, and some of those compounds involved are thiol compounds, and others maybe esters or lactones or other compounds that would be contributing as well. So overall, they're really very odorant. They're very potent. So when we talk about our normal range of sensory for most of these fruity compounds in beer, like esters, we'd be talking about more the parts per billion, which is still a low level. Parts per million would be more on an ester level. Parts per billion would be like a diacetyl level. And we're getting even further down to parts per trillion for thiol compounds. So, the aroma impact of a thiol can be, a millionfold lower than the odor impact of an ester, which is pretty impressive.
Right.
Yeah.
And please.
In beer, we find many different thiol compounds, some pleasant, some unpleasant. The ones that we're going to talk about today are the polyfunctional thiols that are more contributing to fruity character, but there's also other thiols that contribute to off flavors. So we know skunky, 3-MBT, the light stroke character, that is a thiol compound. Other, off flavors or reductive kind of sulfur compounds are also thiols. So talking about more off-putting like rotten eggs or rotten vegetables, like garbage character, that's also thiol. It's not the good stuff we're gonna talk about today though.
That's great. I mean, one of the point that I want to stress out here is that when we talk about thiol, most people are gonna map it with those highly aromatic compounds from the fruit or from the hop itself. But like what you've already touched on, there's the other camp that you really don't want it to have it in your beer because it's very potent too, right? The 3-MBT and
Yes.
the captain and stuff. Okay. Now since the topic of this session is about thiol, so you briefly say about the free form and probably touch on about some bound form. So let's further explore in this arena. So can you talk a bit more like, the form of thiols and how important of those two?
Sure. So basically, biotransformation was kind of a word that people threw out a lot in the industry for a while before we actually had evidence of any of the yeast transforming compounds in the malt or the hops. But this is an example of what we would term biotransformation. It's one compound that's converted by the yeast into another compound, and the compound that is starting in the raw material is actually non-aromatic.
Right.
So it doesn't actually provide any impact in the beer. It's just kind of there and waiting to be freed up and released as aroma by the yeast. So in the bound form, these are conjugated compounds to molecules called glutathione or cysteine, or even some intermediate compounds. The plant that itself actually uses this as a way to kind of detoxify aldehydes that are produced during fatty acid metabolism. And by taking those aldehydes and conjugating them to glutathione, it's able to kind of detoxify them and store them in the plant. So these are just mainly kind of remnants of the plant's metabolism that really are not providing any aroma impact in beer. But with our yeast, there or potentially, other enzyme, capabilities, we're able to convert that bound compound into a free compound, which then creates a really aromatic, passion fruit or guava aroma with some of the compounds, or, even like grapefruit, caddy, rhubarb, as we were talking about. So from converting from bound to free, we're getting really high impact of aroma, but it's also, we have here on the slide, the majority is bound form. And in many cases, the bound form is over 1,000-fold more abundant than the free form. So there's a ton of aroma potential that's kind of waiting there in the raw material. And then, yeast is one way of unlocking that potential and producing aroma. So in our raw materials, we actually have these bound thiol compounds in malt, which is kind of one of those things that was unanticipated. We were mainly looking at how to free up aroma of hops, because we knew there was a lot of bound compounds in hops that could potentially be released and contribute more aroma impact. But we actually found there's quite a bit of, especially what you have here is the 3-MH or 3-SH compound that is really abundant in a bound glutathione form in malt. So yeah, we're finding ways of kind of bringing even passion fruit aroma from the malt itself, which is kind of crazy.
Yeah, it sounds super crazy. This is out of my curiosity. It just pops up, but do you happen to know who or when that people actually found out about thiols in malt?
Yeah, that was more or less, I would say, some of the original work from Laurent Dagan, and we worked really closely with the Lab Nicios to do a lot of our thiol analysis originally. And it wasn't long before we started working on thiols that there was some evidence that the malt was contributing the bound thiol. It might have been maybe in 2019 or 2018 that started to become, learned and kind of accepted that the malt would be a source.
So pretty recent, okay, because it's pretty kind of weird, because you say that it's kind of a response for plant metabolism, right? For forming this bound thiol to kind of detoxify something, if I get it right. And whoever thought that our barley also has that kind of thing, and we can free it up if you have the right tool, right?
Yeah. And it's actually really neat too, because the barley has this bound component, but unlike in grapes, where the bound component is very abundant and yeast is actually freeing that compound. So in like a Sauvignon Blanc wine, we actually knew already that yeast was making this tropical aroma from the bound precursor. Wine fermentation and yeast metabolism in that fermentation is very different from in beer, but we never really had evidence of that thiol component coming out of a beer fermentation. So it just wasn't recognized that the malt was a big source. But then we started to learn a little bit more about it, and I think we'll get into this probably a little bit more in our conversation, but it is really down to yeast metabolism and how they activate the carbon sulfur lyase or beta lyase enzyme. That in wine it works, but in beer it doesn't. So it's really curious, that we kind of didn't know more about that potential, but it really comes down to the yeast's ability to free it.
All right. So, we've already seen here that the majority of thiol in raw brewing ingredients exists in bioform, like 1,000-fold that you mentioned. So I think the next step is to learn the mechanism of how technically can this bound form be liberated. So can you walk us through it?
Of course. Yeah. So in this diagram here, you're just looking at kind of the compound when it's in its bound form, when it's conjugated to cysteine, as it's shown. And there's a carbon sulfur bond there, the beta carbon sulfur bond that's indicated. The enzyme beta lyase is active on that carbon sulfur bond and liberates the free thiol or 3-SH or the SH group. And then instead of cysteine, it's actually making pyruvate and ammonium here. So the enzyme is breaking down that precursor compound into the free aroma compound, or the 4-MSP in this diagram. Okay. So an enzyme is capable of cleaving that bond. That's how the aroma is released, basically.
And the source of these enzymes, like you mentioned before, is available in yeast, but it depends whether which yeast is it, right? Like wine yeast or brewing yeast.
Yeah, so it's less about the yeast the type of yeast that is freeing up the aroma. It's more about the conditions of the fermentation. So in wine fermentation, you become nutrient limited. So a lot of wine fermentations require nutrient additions to go to completion and to be successful. But when the nitrogen in the fermentation becomes depleted, the carbon sulfur lyase or the beta lyase enzyme is then expressed. So it doesn't happen in wine until those nutrients are depleted, but in beer, the nutrients never become low enough to activate the beta lyase enzyme.
We feed them too well.
Yes, exactly. We're too nice to our brewing yeast. They're always, in almost every fermentation, we're in nutrient excess. So especially in terms of nitrogen. So there's very few circumstances in beer where we have to supplement with nitrogen. So in that case, the yeast is kind of spoiled. Never has to turn on this pathway. The pathway is actually meant for the yeast to be able to uptake and synthesize its own sulfur-containing amino acids and scavenge kind of and be able to metabolize them on its own. But it never really happens in beer because they're always present. So in the case of even if you were to put a wine yeast in the beer fermentation, we did this in our early experiments. The wine yeast does not release thiols any better than the beer yeast. It's really the context of that fermentation, the nutrients that are present, that allows the yeast to activate the beta lyase enzyme.
Got you. Like the first time that I saw this article, probably 4 years ago, about the study of thiol and the bioform and freeform, it was kind of a whole new world for, it has never been, done before in the brewing industry, unlike in the wine world, right? The wine industry, they study thiols for quite some time ahead before us. And this is just the quick question, like what sparked your attention to look into the bioform of thiol and try to find ways to release it at the beginning?
I mean, I definitely, I love wine as well. I'm a beer kind of by, what my profession is, and also by, how I started drinking beer before I started drinking wine. I'm really more of a beer drinker, but I do like wine as well. The inspiration came from tasting Sauvignon Blanc and just knowing these tropical aromas are wildly impactful, and it was something that I truly liked. It was something that just kind of struck me as, wow, the yeast actually can produce these compounds. This is crazy. And it really inspired me to be working on it. So it really came from, the experience of drinking Sauvignon Blanc that I became interested in finding out more about how yeast could be active on those compounds. And then when I was studying early on, I was looking at a lot of the wine research. So that was part of like the groundwork and foundation that allowed us to get, started on the project and to kind of stand on a lot of that research that had been done before us. So it was really impactful to have all of that work done in wine for us to even get started in beer. But we did have to learn a little bit more about how it was working in beer to understand, to do what we were doing. So in our early fermentations, we were actually experimenting with beer, wine, yeast hybrids. So we were mating beer yeast and wine yeast to generate strains that would have high beta lyase activity. And that was kind of our first indications of when we were doing the fermentations with beer yeast, wine yeast parent strains, and then our hybrid strains. We weren't observing that aroma. That Sauvignon Blanc character was not coming through in the beer fermentations. So that was our first hint that we weren't getting appreciable amounts of those compounds. And actually, we were sending out those fermentation samples to Nicios and seeing that we were only getting a very minor amount of thiol release, which wasn't really impacting sensory.
Right.
Yeah, so we kind of pivoted from there and really looked into the opportunity to engineer the strains, since we weren't getting enough of the thiol release from the hybrid strains.
Wow. So basically, it literally came from a simple question. It's like, how can the Sauvignon Blanc wine fermentation, the final result wine in the glass has this kind of really nice aroma, and how can it be projected to the beer world? And you just start, digging down to this hole and try a bunch of stuff, which is kind of a way to lean on what we're going to discuss next with the current approach at Omega Yeast to liberate and find a way to make our brewing yeast be able to activate or express this beta lyase gene, right? So please talk about it.
Of course. So then without the release from traditional strains or wine beer yeast hybrids, we went to an engineering approach. So in this slide, it kind of shows you a little bit of CRISPR-Cas9, a little diagram of the complex of the DNA and the Cas9 guide RNA complex where our editing event occurs. But we use this gene editing technology to introduce an active form of the yeast beta lyase IRC7, or in another set of strains, an active form of a bacterial beta lyase, PAP-B. So by using this really targeted gene editing technology, we were able to go in and very specifically introduce those changes into the yeast and see what effects they had on fermentation. So our first experiment found that by activating the yeast IRC7, we could get that sensory level of thiol coming through in fermentation. And it was, even with the highest expression of the yeast IRC7 enzyme, we were getting to levels of 10, maybe 20 times sensory threshold. So we were observing it in the beer, and it was coming through with that passion fruit aroma. Right. And then to go even further, we actually used another beta lyase enzyme that was more active. So it produces, a considerably higher level of thiol. And that's that PAP-B enzyme from bacteria.
Mhm.
And by introducing that, we were about 200x above the threshold of sensory. So
Mhm.
in the early studies, we were working primarily in the yeast themselves, just trying to turn the pathway on because we knew it was turned
Right.
off in beer. And that was
Mhm.
pretty successful with our first strain, Cosmic Punch. But then
Right.
later, we wanted to do, basically introduce even more aroma, and found that this PAP-B enzyme was another approach. So we have two different ways of activating these beta lyase enzymes, and they have two kind of different applications. Some people like a modest amount of thiol, and others people like to
Mhm.
be really just
Extreme.
extreme, and it kind of fits into different approaches to brewing as well. If you want to be very intense overall, and you're doing a high
Right.
amount of dry hop, and you're doing a double IPA, then it helps to have
Mhm.
a lot of thiols produced, because those have to
Right.
stand up to the other compounds that are present and actually
All right.
be perceived. Because we can talk about this a little bit after too, but
Yeah.
what we've learned about the thiol sensory perception and preference, and whether people detect the thiol at different levels, is part of the whole equation too. And it really does make these strains pretty, it's useful to have a low level and a high level for the different types of beers that people want to make.
Got you. So it is very interesting to actually learn that. I'm not really sure, like, is the use of, or the option to go with the engineer yeast trial, is that the first on your list to try to kind of tackle this beta lyase liberation, or it's like, are there any other ways that you kind of work before? Because you mentioned that you did the hybridization, but it doesn't really express, right? And is this engineered yeast is like the second choice and say, hey guys, let's just try this way out, or okay.
Exactly. Yeah, I think there's quite a bit of research even in beer yeast currently that's suggesting that beer yeast are capable of freeing thiols. But when it comes down to sensory impact and thresholds of these compounds, if one of the best strains for producing thiols are the types of yeast that have been are Kolsch strains and lager strains. And we know we brew these beer styles very simply.
Right.
And we try to, execute the cleanest beer with these yeast. So if what the main use of these yeast is in producing these very clean styles is not leading to any thiol aroma,
Right.
then we
Right.
already know those strains in their current
Best condition.
how we use them. They aren't active in releasing thiols. The amount of thiols they are releasing doesn't impact the sensory, or impacts the sensory in a way that doesn't come through as passion fruit. There might be other influences that kind of come together in the overall aroma, but the aroma isn't perceived as passion fruit.
Okay.
So when we think about traditional yeast or beer yeast hybrids, the level of thiols that are produced may be measurable, but they're not producing passion fruit aroma in a beer. If that makes sense. Yeah.
Right. Okay.
Yeah.
Make a lot of sense.
Unless you've had a passion fruit forward lager, which would be really cool because I want to try it. I've tried them with these engineered strains, and they're pretty intense and exciting, but I've never experienced that type of aroma out of a traditional lager.
Right. For real. Yes. Yes, you're right. And for the IRC7 gene, I have a question, like how difficult it is to identify genes that response for, say, beta lyase, like this one, or the other genes for haze. How difficult is the process to look into this specific genes?
Well, that's actually a really good question. When we started in our R&D here at Omega, we kind of took a early approach at building out our bioinformatics capabilities, which means that we can look at whole genome sequencing of yeast strains, and know more about the structure of the genome and what types of mutations they have and how those mutations might impact fermentation. So we kind of, to do the work we do, we have to have big component of that being really foundational research and, kind of similar to the work I've done in graduate school, building out our capabilities and understanding the whole sequence of the genome to be able to apply it to beer. So yeah, we've done quite a bit of work there. That's how we came across this hazy 1 gene is by really getting into the genetics and with an unknown genetic component, we were able to link a phenotype to a specific gene. But with the beta lysis enzymes, because we had a lot of this foundational information in wine, those genes were already known. Alleles that were active or inactive were kind of already known. So our approach just involved putting the active allele into a brewing strain and making sure that active allele was present and expressed at a high level. So part of the work was already done. So we had a little bit of an easier time with the thiol project we did on the haze project. But yeah,
I mean, like it's kind of a big puzzle for. I mean, like, I really like biology, biochemistry, but this is kind of, hey, we have to do a lot of research, which gene responds to what. That's kind of big thing, and it's very cool to see how it can actually translate to the, I would say, finished beer somehow in the most simplest way.
Yeah, and I don't know, even when you look at brewing strains, we have a very complicated genome because they've been, domesticated, and, we have 4 copies of every gene, and there's a lot of these aneuploidies and other things that make it a little bit more complicated. Brewing strains are a little more difficult than just native Saccharomyces cerevisiae. But if we think about it, we still only have 6,000 genes, and it's still a pretty simple genome. So it's kind of it's nice to be in an organism in a simple context, because then you can apply some of the more advanced techniques to understand something very simple. But those techniques are applied to human genomes, so they're very robust. So we kind of work in a system that, puts a little bit of a advantage to us and allowing us to do a little bit more with a simple genome.
Right. That's awesome. So last one on this particular subject, can you lightly touch on the principle in a nutshell of the CRISPR-Cas9, like what it is? I understand that it's the Nobel Prize won method that get famous in the past couple of years, right?
Yeah, it is it's super useful for us. We've tried a lot of ways to do it without CRISPR, and honestly, there's not anything comparable.
It's like a bingo, right? Just check the box.
Exactly. So it's kind of like an advance to going from, a typewriter to a word processing program or software. You're able to the enzyme and the RNA complex. The RNA is hybridizing to a specific site in the genome, the guide RNA. The enzyme hones into that guide RNA DNA complex and then does a specific edit at that site. So if we're thinking about it in the context of a word processor, you would basically be using the find function to locate the word that you wanted to edit. You have a cut and even a copy and a paste if you want. So by using the CRISPR-Cas9 tool, you have truly the ability to go to anywhere in the genome and make a specific change. The change can be, to remove a gene, to add in a gene, paste a gene, or, even just do a single change of almost like a spell check to change one letter to it. So, it's just super powerful. Before we did have options to engineer brewing strains, but it was just really the insert function. We didn't really have the editing function. So now that we have CRISPR-Cas9, we can go in and do very targeted changes anywhere in the genome, which is really cool. Yeah. So one of the examples that we did at Omega, we took phenolic Hefeweizen strain and made a single nucleotide change, 1 in 12 million nucleotides in the genome, one change, one base pair. And it went from a phenolic strain to a non-phenolic strain. And that same change is what is already in our American Ale strain. So the non-phenolic character of American Ale is linked to that one base pair change. So we can use that naturally occurring change and introduce it and I think that the power of that is that it really is analogous to breeding, because you're not introducing any foreign DNA or changing any DNA in the organism that wouldn't already be there in nature. So that's why CRISPR-Cas9 has also been more globally kind of turning over as a widely accepted kind of technology for gene editing, because it is analogous to breeding in many contexts, and many nations that are working towards their regulatory process have recognized that. So it's just a nice, I guess, tool that made some of these these opportunities to engineer yeast or plants or animals even more widely accepted and easier.
Yeah, it's pretty like a jaw-dropping technology in these decades.
Yes.
In the realm of gene engineering technology.
Yeah, and we can do a sequence of a strain that we are wanting to edit, and then we can edit the strain and do a sequencing of the strain after editing and compare them and be sure that we really had that one change. So it's that specific that you can even confirm and verify that the change was specific and you haven't had any other off-target events, which is how people are demonstrating this is so simple and really akin to the kind of the breeding concept.
Right. And I really like your analogy for, the find function. You can just find the spot anywhere you want, replace, cut and do precise editing or engineering to yield a result. So basically the answer that Omega Yeast currently attack or tackle for this bound thiol is through this engineered yeast with the CRISPR-Cas9 technology. You use to select the gene, the IRC7 and PAT-B, to create the offspring for this thiolized yeast, which is the next slide that I'm gonna ask you to just run us through or introduce these four strains to all of us. What's its up and what's its consideration that brewers supposed to know.
Oh, absolutely. Yeah. So as we were talking about the two different enzymes that we've used, the IRC7 or the PAT-B. IRC7 is the yeast enzyme, so it's active and it produces a considerable amount of thiols, but it's not producing to levels that are screaming in the beer. It's more a delicate amount of thiol, an enhancement of thiol, similar to like lower dry hopping rates. It's if you were to compare the two, like a hop that has a modest amount of thiols would produce this level of thiols that Cosmic Punch is contributing. So I guess in a sense, if you had a more of a West Coast IPA style that was using Pacific Northwest hops with a good amount of thiols, but not extreme amount of thiols, you might expect to see Cosmic Punch level of thiols in those beers. But if you're using a New Zealand Rawaka Nelson Sauvin hop, which has screaming amounts of thiols, that is more what you'd be getting from Heliogazer. So kind of similar to those hop varieties where you have low and high thiols, these yeasts are producing low and high amounts. So if you're going for tropical aroma, you want a tropical IPA, Heliogazer is your option. If you're going for more aroma impact and complement to kind of hop aroma, then Cosmic Punch is kind of the better option here. And it really just comes down to the enzyme that we expressed in those strains. So internationally, if we were to be able to bring thiolized yeast to other countries, the easy option would be the IRC7 because it's coming from the yeast itself. And that's usually, more widely accepted globally as an engineering or editing approach. But putting in a bacteria enzyme into yeast, it's only a few countries that allow that or have deregulated that. So in the US, we kind of are in a sweet spot where we have access to be able to, introduce
play around
things in the market. But Canada, for example, we sell IRC7 because they've opened up the possibility of those yeast alterations, just what we call cisgenic alterations.
Oh, okay. Okay.
Yeah. So I'm mentioning this because in the global sense, there are probably, countries that will become more permissive to these technologies. And if we see that turnover, it'll likely be with the Cosmic Punch strain where
Mhm.
it's the modest amount, and then the next to come would be these Pat B strains.
The Pat B strains.
Mm-hmm.
So there are four. Currently, there are 4 strains at the moment, right? Or more that are okay.
Well, so we have the we have a couple more that we engineered with Pat B. We have a Bossa Nova strain and a Little Dippa strain. So Conan and Voss that have been engineered to have thiol expression as well. So we can plug and play. That's the kind of benefit of this technology too. We can plug and play this into any brewing strain background. We can really target that specific change in any of the brewing yeast we have.
Yeah, I mean, like back like about 2019, I guess that was the first time that I had my hand on the Cosmic Punch, and I homebrew with it. And I mean, like I did some studies, read through all your research and how to best use them at that time. And I remember that the result, I was like, holy, this is like something in another level already, like those kind of passion fruit and guava that I get with, the just simple base grain and simple hops, nothing crazy. And probably like less than a year or half a year later, you guys released Star Party. And I was like, I need to try that. And after that batch, I was like, oh, this is like, if I thought Cosmic Punch is just here, I mean, like here, the Star Party is like the one shown in that graph is like throughout the roof.
Yes, exactly. So in when we talk about the Sauvignon Blanc aromas that we get in a New Zealand Sauvignon Blanc, the mounts are really up in there. The 12 ppb that this is measuring here is similar to what would be found in a Sauvignon Blanc wine. So you're getting that level of intensity. And if you're dry hopping with like 2 pounds per barrel of a Nelson Sauvignon, you'd be hitting that level of thiol intensity too. Wow, so we're getting a lot of thiol intensity with these strains. But again, when it comes to recipe design, it's Cosmic Punch might be overshadowed by other, if you have high dry hopping rates or other aroma, it might be masked a little bit. It might not stand out as much. But in a heliogazer, you're really still getting passion fruit aroma in the end beer, even with the high dry hop amount.
Yeah, and I was a bit skeptical back then when I found the study that shown that barley or grains has bound thiol, and you don't really need to rely on hops. And I was like, okay, let's just try that. And I was proven wrong. I mean, like myself.
Especially that's like when we were using these strains initially, and we were just trying just wort without any hops. It was tasting like Sauvignon Blanc wine. And and when you walk into the facility here at Omega Yeast in a strain with a thiolized, like a Pat B or IRC7 strain is being produced, you can smell the aroma in the facility. It smells like passion fruit. So like potent. At those moments, we were very aware that this is all coming from the barley. There's no need for any additional precursor in those Pat B strains. Like the barley is enough to support all of that. And actually, that's an interesting point to make. We talk about barley, but actually wheat and oats contribute very little precursor. So if you were to ferment 100% wheat, you wouldn't get that same aroma released out of the wheat. So it really does rely on that barley, and even the barley variety, the malting process, all of that plays in. So who knew we have that concept of terroir and also the agronomics and the influence of malting that changes what we can get out of it as in terms of thiol aroma.
Right. Okay, that's great. I know we've come this far from that simple set of questions at the beginning of this session, and we are now at the tail end of it. Right now we understand what thiol is. Its form, the bound, the free, how important of the bound to the brewing world, and the way that you can liberate them, the use of engineered yeast and the technology behind them. So I think the last part here is to try to understand these new tools in the brewer's tools block. Some of the ways that you can manipulate, dial up or tune down the thiol expression in your final beers. So can you share with us with some tips and tricks of doing this, the way to control or manipulate the thiols?
Yeah, of course. Yeah, some of this is, well, we were learning as the brewers were learning, and this was all new to everybody. So in our early experiments with Cosmic Punch, we really wanted to coax more of this aroma out because it was reaching the amount where it became obvious, but we wanted it to stand up to a dry hop beer or something that was already intense. So with that, we were working through strategies of maximizing a thiol aroma. So with, again, focusing on your grist, the more barley you have, the more barley precursor is present. So that's important to think about your grist composition overall. We worked through some concept of mash hopping and also adding wine grape precursors. So the Phantasm product out of New Zealand was actually taking the grape skins of Sauvignon Blanc grapes and pulverizing them to use in beer. And it contributed a high amount of precursor. So in cases where you were trying to maximize thiol aroma, you were wanting more precursors. So those were the two strategies, is either introducing them with mash hopping or introducing more precursors with this Phantasm product. And then, the other impact we found that the more the vegetal material, the more the plant material, maybe polyphenols were kind of complexing and reducing some of the aroma impact of those thiols. So we were finding that, less of the vegetal material was actually better for maximizing thiol aroma as well. Then we moved on to, introducing our pet B strains. So maximizing thiols wasn't as critical anymore because you already kind of maxed them out with the yeast. So some of those techniques aren't as necessary with the Pet B strain. But so brewers kind of look for both options. They want to either maximize or they want to just have a thiol aroma that's part of the equation but doesn't dominate. So to lessen intensity, you would stop with the mash hopping or any additional precursor, kind of lower kind of the amount of barley in your grist. So incorporate some of the other adjunct, wheat or oats, to kind of lessen the amount of barley precursor that's present. And also picking the IRC7 strain over the Pet B strain, giving a moderate amount of thiol release versus the very high level of thiol release. So a lot of play here with optimizing, depending on what type of beer you want to make. Also, it comes down to personal preference a lot of the time. You as a brewer might not love high amounts of thiol. It might kind of be an off note for you. I have people on my team here at Omega who perceive thiols in high levels as sulfury, off-putting aroma. And in that case, they don't like thiols over the top. They want a moderate amount. They don't want them to dominate. So in aroma quality of your impression of thiols, it's very different between everybody tasting it. We have individual preference, and it all comes down to how our sensory is wired. It comes into play here more than you would think. It's either, I guess, in the grand scheme of it, the individual's ability to detect thiols can change. So you might not detect them at low levels. So every individual has a different threshold of how much thiols they would be able to perceive. The overall attributes that thiol brings to a person can change. So some people only get passion fruit. Some people get a little bit more of the sulfur component. So like any compound in beer, there's haters and there's absolute fans of these compounds.
The fans of it,
Phenolics or bitter or sour. There's always a spectrum of who prefers or maybe dislikes those at different levels. So something just to consider when you're designing recipes with these is there's definitely kind of a wide range of thiol amounts that you can get in the beer, and also that amount may fit a different style more appropriately. So pale ale, just moderate amount of thiols, might be an IRC7. You might want the Cosmic Punch to be your strain in that. But if you're going for really intense tropical IPA, then you'd want these peppy expressing strains like a Star Party or a Heliogazer.
Yeah, so it just all boils down to, I mean, as for the brewer, like what kind of beer you want to make, what the end result that you look for. Are you looking for those that are super expressive and gonna knock people's socks off with a surprise, or you want something that harmonize well with, at the base level, and you have this set of tools to play around, right? The hop selection, the yeast strain selection, the process selection, and just cook all this thing up to, try to brew the best beer possible.
Exactly. And if anybody out there is just experimenting with these strains like you did, you can just do a simple recipe. Just try out the RC7 and the Cosmic Punch and Heliogazer as a comparison to the parent strain, which is the London Ale 3 strain. If you're doing that comparison, you're going to really see how thiols impact the finished beer at low, moderate, high levels. And also, if you just want to experiment and play around, you can actually find food grade versions of the compounds to try to just do a little bit of sensory yourself and understand what you perceive and what amounts you like, and then target your recipe to those amounts.
Okay, got you. Thank you for sharing this tip and tricks to all of us. It's very valuable. I have probably one last question before we close this session out. It may be a little bit, I'm say dumb question, but this beta lyase enzyme, we don't have the one that is kind of been synthesized to the liquid form yet, right? Like, those other enzymes that we can use exogenous. No. Okay.
Yeah, nothing in the market, but I don't think we have a good understanding, since these are all expressed in the yeast. They are in a very different environment than they would be in the beer. So to date, nobody has anything available that works in beer.
Right, because I understand that we have the what's the other one? The one for biotransformation. Yes, glucosidase. Yeah. Okay, yeah, but not for the beta lyase yet. Okay.
Yeah. So bound terpenes we can free up with the beta glucosidase, which that is a commercial enzyme that's available, but there isn't anything available for thiols. Okay,
so can I put it this way, that it's the current limitation of our technology at this moment. The best choice now is engineered yeast. Okay,
that's right. Yep.
Go for the yeast first.
Yeah. It is, there's definitely efficiencies when the enzyme's in the yeast. So we have other examples of where we've engineered in the ALDC enzyme to yeast, and it avoids any added effort or expense of adding the exogenous enzyme. So there's cases where building in these capabilities to the yeast actually is a easier and more cost-effective option.
Okay, I understand.
Yeah.
All right. We are at the goal line. Thank you. And to close the session out, Laura, do you have any final says about this topic?
I think it's been one of those projects for us that is just been more enlightening than we ever thought. We love learning. We love diving into these big questions that maybe we thought we knew a little bit more than we actually did. So the whole path for us has just been really fun to understand more about how it works in yeast and how it works in beer. And if there's, I guess, anybody out there, it's just keep your curiosity and never think about things as kind of true until you start testing them and understand a little bit more about the limitations of what we know, because there's always more to learn.
That's great. As a follower of this study and research, I'm sure that for me, I find it very helpful, and I also enjoy learning it, along the way. And I'm pretty sure that what happened behind the scene, if you skip all the hectic part, the head scratching part, you guys also have a lot of fun time doing this.
Yes, we do. We definitely do. I think some of those first moments of getting a beer that you actually, whoa, it worked, is always, or walking into the facility on the first time we ever grew it up at scale and smelling the aroma. Like those are the
It
moments
works.
that get you definitely, you know, really just thrilled about the science and seeing that really come into practice. So yeah, it's been really fun for us too.
That's cool. That's excellent. And this is where we will end the session. So Dr. Laura, thank you so much for guiding us down this sciencey road today for this biothiols topic. I truly enjoyed our conversation. Thank you.
Yeah, same. Thank you for having me.
Okay, it's time to send you back to your day now. Thanks again. Take care.
Thanks. Bye.
Thanks. Bye-bye. All right, here you have it, biothiols and its liberation capability from the engineered yeast. If you happen to have a chance to try the beer that, being brewed with this engineered yeast, thiolized yeast, just, give it a try. The worst thing that can happen to you is you're not gonna like it, but just move on. At least you just give it a try. But trust me, if you ever have a chance to have a sip of it, even it is too overpowering for you, I think part of your brain will kind of process it and see how you can turn this amazing idea or amazing taste or amazing aroma into something that you like on your next brew or on your next journey of brewing. So yeah, just keep learning, keep your eye open, and just look forward for the new things that happens in our brewing world. That's all for today's session. The Good Burp session will be back in no time, so stay tuned. Until next time, cheers and sawadee krap.